The BBSRC has three main themes to its strategic plan for research. One of these is the basic bioscience underpinning human health. That's where Babraham's science is focused. Unless we understand the basic bioscience which underpins human health, then we will not be in a position to be able to understand what goes wrong in human disease. So Babraham's research is split into three major themes. Firstly, there's the cellular signaling grouping, secondly, the immunology grouping, and thirdly, epigenetics and chromatin. In the signaling program, we're understanding how acute responsiveness occurs, for instance, in the way in which a body deals with infections. That leads into the immunology, where we're understanding how the immune system develops and how it changes during aging, for example. Whereas within epigenetics and chromatin, we're looking firstly at developing organisms and secondly at how the body has adapted to changes in its environment.
Epigenetics are modification or changes to the DNA and the chromatin in the nucleus, which make the genome experience the environment in a particular way and talk to the genome and make it function, make the organism function in a particular way. If you imagine that there is about 200 or 300 different cells in the human body, they all have their own unique epigenetic information and an epigenome, as we call it, which is uniquely characteristic of each cell type. So there's liver cells which have a particular epigenome, there is brain cells which have another type of epigenome, and the epigenome in essence instructs the genome to only use a particular subset of all the genes that it has in order to confer identity, be a liver cell, be a brain cell, on the particular cells in the body. For example, we know the colleagues here at the Institute have discovered that a particular class of genes that epigenetically regulated controls the amount of fat that the body acquires during development and after birth and later on. And so it's possible that this type of epigenetic regulation underlies the development of obesity, for example, later in life, and perhaps also diabetes.
Babraham has a number of technologies which facilitate its work. Firstly, there are mass spectrometry methodologies which allow us here not to just look at proteins, as in many places, but also at lipids, which are critically important in signaling processes in cells. Secondly, we have specialized systems such as fluorescence activated cell sorters, which allow us to isolate particular populations of cells, for instance immune cells, for study.
Babraham is at the forefront of epigenetics research in Europe. Is one of the largest centers that carries out this type of research. And the technology that we've been able to develop here and build up through investments by BBSRC, MRC, and also the University of Cambridge has really put us at the forefront of this kind of epigenomics field worldwide.
Our imaging facility has a number of microscopes. Particularly the confocal microscopes can be used to identify the localization within a cell of various proteins or genes. This technology has been developed further recently to allow the examination of these cells under live conditions. This means that we can look at the movement of proteins, for instance, within a cell when it's been stimulated. A good example of this is a neutrophil which is responding to an infection. We can look at the movement of signaling proteins within these cells in response to the infection and see how that changes in the presence of various pharmacological inhibitors, therefore allowing us to investigate the disease processes. Links with companies is extremely important for Babraham's science. That is because whilst the research has its own intrinsic importance, at the end of the day it has to have a benefit for society. We have a number of links with the companies in our incubator on site, of which there are 32 at present. Some of these are spin-outs from Babraham's science, and others have chosen to locate here where they can, amongst other things, interact with and collaborate with the Babraham scientists.
Crescendo is a new biotech company established at the beginning of 2009, aiming to make new antibody-based therapeutics. The advantages of antibodies as therapeutic agents is that they're highly specific and they don't have the unpredictable side effects of small molecule drugs. At Crescendo, we're trying to develop antibody-based fragment therapeutics, which have those advantages, but also some of the advantages of small molecules. In other words, topical delivery, no need for injection. The science that's the basis for Crescendo has been occurring over about 10 years here at Babraham, and it's been pioneered by Drs. Marianne Bruggeman and Mike Tausig. The advantages of being based at Babraham are that we live in a very rich scientific environment, and we're constantly tapping into the scientific expertise of the Institute. There are about 20 or 30 other companies based at Babraham, and that makes it an exciting environment to be in.